Hi, and welcome back to another A-Level biology video. We're starting a new series called Biological Molecules, and we'll be talking about the chemistry behind life. In this video, we'll be talking about carbohydrates. The carbohydrates are molecules that are made out of carbon, hydrogen and oxygen. They can be polymers, which, as you know, means a long chain of repeating units called monomers. Examples of polymer carbohydrates or polysaccharides are starch, which you find in pasta and rice and potatoes and bread, glycogen, which we find in our own bodies as an energy store, and also cellulose, which is found in plant cell walls. Carbohydrates can also exist as monosaccharides, so that's single sugars like fructose, galactose, and of course our very favourite in A-level biology, glucose. In fact, we need to know about glucose in quite a bit of detail. So let's get started.
Glucose is a hexose sugar, which means it contains 6 carbons. There are 2 isomers of glucose, alpha and beta. Let's look at alpha first. You can see the 6 carbons are more or less in a ring. This is alpha glucose, and it has both of its H groups pointing in the same direction and both of its OH groups on the sides pointing in the opposite direction. Now we will compare that to a molecule of beta glucose. Beta glucose is exactly the same as alpha glucose in every way except that one of the OH groups has swapped places with one of the H groups. So now one OH group is pointing up and the other OH group is pointing down.
So to summarise, alpha and beta glucose are isomers, which means they have the same number of atoms, but they're just arranged in a slightly different way. Why does this matter? Well, it does affect the structure of the polysaccharide chain that they form when they join together. And we'll come to that in a moment. First though, we're going to look at how 2 monosaccharides join together to form a disaccharide. This is an extremely important reaction to learn.
Disaccharides are just 2 monosaccharides joined together. Sucrose is an example of a disaccharide, as is maltose. Maltose is 2 glucose molecules joined together. Here we have 2 molecules of alpha glucose. We remove a molecule of water and a glycosidic bond is made. Because we're removing a molecule of water, we call this a condensation reaction. You need to be able to draw out this reaction, and when you do, please don't forget to include the molecule of water as the other product. Just because it's small doesn't mean it's not there. Here are the 3 disaccharide reactions you need to know, and they're all glucose plus another monosaccharide. We've already done glucose plus glucose makes maltose. Then there's glucose plus fructose makes sucrose. Fructose is the main monosaccharide found in fruit. And finally, glucose plus galactose makes lactose. Lactose is the disaccharide found in milk.
So we've seen how removing one molecule of water can join 2 larger molecules together. So does that mean adding a molecule of water might split them apart? Well, the answer is yes, absolutely. It's the opposite reaction. And we call that type of reaction hydrolysis, which literally means splitting using water. Okay, so how do we test a food sample to show that it has sugar in it? Well, at GCSE, we should have learned that you add Benedict's solution and heat it up to about 90 degrees. And if it turns from that light blue colour to a brick red colour, that means there's sugar present. Well, it's not quite that simple, unfortunately. What it does show, if there's a colour change with Benedict's solution, is that this food sample contains reducing sugars. Reducing sugars include all monosaccharides, so galactose, glucose and fructose, but they don't include quite a number of disaccharides, including sucrose. So sucrose is definitely non-reducing, but it's still a sugar. It's the sugar that we put in our tea. So what we need to do is add an extra step to the food test for sugar.
First, we'd carry out the standard Benedict's test. If the result is positive, then we know that we have a reducing sugar. But if the result is negative, there might be some non-reducing sugar that we do not know about. To test for this, we get a fresh sample of the food and we heat it with hydrochloric acid. The acid and heat break down any disaccharides into monosaccharides, which, as we know, are always reducing. Next, we neutralise the solution with sodium hydrogen carbonate so that the Benedict's test will work. So then we can carry out the Benedict's test just as before. If there had been any sucrose present, it would have been hydrolysed into... That's right, it would have been hydrolysed into glucose and fructose.
And finally, we come to polysaccharides. Polysaccharides are long chains of monosaccharides joined together by glycosidic bonds. They are usually used as energy storage molecules or as structural components in cells. We need to know about 3 polysaccharides in detail, and that's starch, glycogen, and cellulose. First of all, starch. Starch is the energy storage molecule in plants. As you know, in photosynthesis, plants make an awful lot of glucose. However, glucose is soluble, and so it can affect the water potential of the cell, leading to damage to the cell by osmosis. So as soon as the glucose is produced, it needs to be converted to starch in order to store it, because starch is insoluble, which means it won't affect the water potential of the cell.
There are 2 types of starch, called amylose and amylopectin. Amylose is a long, unbranched chain of alpha glucose that exists in a coiled structure. This makes it very compact and good for storage. Amylopectin is a long, branched chain of alpha glucose. The branches increase the number of endpoints on the molecule for enzymes to break the glycosidic bonds more easily. This means that the glucose can be released much more quickly for respiration, for example.
On to animals now, and we need to store our glucose somehow as well.
We store our glucose in long, branched chains of alpha glucose called glycogen. So glycogen sounds very much like amylopectin, except that it is even more branched. This means that the glucose can be released even more quickly when energy is needed. This is more important in animals than in plants.
Glycogen is also really compact and, of course, insoluble, which means it's excellent as an energy storage molecule.
And to round off polysaccharides, we will return to plants. Plant cell walls are made of cellulose. Cellulose consists of long, unbranched chains of beta-glucose. When beta-glucose molecules join together, there's none of this coiling business, no branching. The chains are completely straight and they lie next to each other with hydrogen bonds between the chains. Hydrogen bonds are very weak compared to covalent glycosidic bonds that hold the individual glucose molecules together in one chain of cellulose. But there are so many chains of cellulose next to each other that it creates this cellulose fibre, which has a very high tensile strength due to the many hydrogen bonds causing them to stick together. So this makes them perfect for plant cell walls.
So that's it for this video. I hope you've learnt something about carbohydrates, and to check your knowledge there is a downloadable worksheet in the description below. Please do like and subscribe if you found it useful, and I'll see you in another video. Thank you for watching.